The human body is a complex and wondrous machine composed of trillions of cells working together in perfect harmony. But here's a fascinating fact. These cells, despite their diversity in form and function, all originate from a single fertilized egg. This remarkable transformation from a single cell to a multitude of specialized cell types is the process we call cell differentiation. Imagine a symphony orchestra with its diverse array of instruments, each playing a unique melody. Similarly, our bodies are composed of various cell types: nerve cells transmitting electrical impulses, muscle cells enabling movement, and blood cells carrying oxygen, each performing a specialized role. How does this incredible process unfold? The answer lies within the intricate world of genes, molecules, and signaling pathways that orchestrate this cellular symphony. Understanding cell differentiation is not just a matter of scientific curiosity, it holds immense promise for understanding human health and disease. The blueprint for life resides within the DNA molecule, a vast library containing genes, the instructions for building and maintaining a living organism. These genes hold the code for proteins, the workhorses of the cell, carrying out a myriad of functions. But here's the catch: not all genes are active in all cells at all times. Think of it like this. Although every cell in your body possesses the complete musical score, only specific instruments play at any given time, creating the symphony's unique melody. Similarly, different cell types express different sets of genes, leading to the production of specific proteins that define their structure and function. Let's take the example of muscle cells. These cells are responsible for movement and require large amounts of contractile proteins such as actin and myosin. Consequently, muscle cells express genes encoding for these proteins at high levels. This selective gene expression is the driving force behind cell differentiation. Understanding how gene regulation orchestrates cell fate is crucial for comprehending the development and function of multicellular organisms. While DNA provides the blueprint for life, the way these instructions are interpreted and executed can vary greatly. This is where epigenetics comes into play. Epigenetics refers to heritable changes in gene expression that occur without alterations to the underlying DNA sequence. Imagine a musical score with annotations and markings that influence how the musicians play the notes. These annotations don't change the notes themselves, but they alter the interpretation and execution of the music. Similarly, epigenetic modifications act like these annotations, influencing how genes are accessed and expressed. One of the key mechanisms of epigenetic regulation is DNA methylation. This process involves the addition of a methyl group to the DNA molecule, which can act like a dimmer switch, turning down the expression of a gene. Another important mechanism is histone modification. These epigenetic modifications are influenced by a variety of factors, including environmental cues, lifestyle choices, and even aging. Cells don't exist in isolation. They constantly communicate with each other, exchanging information and influencing each other's behavior. This cellular communication, mediated by signaling pathways, plays a critical role in guiding cell differentiation. Imagine a group of dancers responding to cues from their instructor, coordinating their movements to create a cohesive performance. Similarly, cells receive signals from their surroundings, which activate specific signaling pathways within the cell, ultimately influencing gene expression and cell fate. These signals can take various forms, including hormones, growth factors, and even physical interactions with neighboring cells. Once a signal is received by a cell, it triggers a cascade of molecular events like a relay race, transmitting the message from the cell surface to the nucleus, where it ultimately influences gene expression. Two prominent signaling pathways involved in cell differentiation are the Wnt and Notch pathways. Disruptions in these signaling pathways can have profound consequences on development and disease. From seed to sprout stages of cell specialization. Cell differentiation is a gradual process, each stage marked by increasing specialization. From a single fertilized egg to specialized cell types, it's a remarkable journey. Think of a seed transforming into a plant. It begins with potential, developing into roots, stems, leaves, and flowers. Similarly, cells progress through different potency levels during differentiation. Section 6. A case study in specialization, the making of a muscle. Let's delve into a specific example to illustrate the intricate process of cell differentiation, the formation of muscle cells, also known as myogenesis. These remarkable cells, responsible for movement, are a testament to the power of cellular specialization. The journey begins with mesodermal progenitor cells, multipotent cells residing within the mesoderm, one of the three germ layers formed during early embryonic development. Under the influence of specific signaling molecules, such as those belonging to the WNT and SHISH or sonic hedgehog pathways, these progenitor cells commit to the myogenic lineage, setting the stage for muscle cell formation. Once committed, these myogenic precursor cells, known as myoblasts, undergo a series of carefully orchestrated events. They express specific transcription factors such as MyoD and MYF5, master regulators of muscle cell differentiation. These transcription factors act like molecular switches, turning on the expression of genes encoding muscle-specific proteins, such as actin, myosin, and troponin, the building blocks of muscle fibers. Myoblasts then embark on a remarkable feat of cellular cooperation, fusing together to form multinucleated myotubes, the precursors to mature muscle fibers. This intricate arrangement of sarcomeres gives skeletal muscle its characteristic striated appearance and enables the generation of force and movement. Section 7. Differentiation in action, implications for health and disease. Cell differentiation is not just a fundamental process in development, it plays a crucial role in maintaining the health and function of adult tissues. Throughout our lives, tissues undergo constant renewal and repair, relying on stem cells to replenish damaged or aging cells. These stem cells, residing in specialized niches within tissues, maintain their ability to differentiate into specific cell types, ensuring the continued function of the tissue. However, this intricate process can go awry, leading to a myriad of diseases. Cancer, for instance, can be viewed as a disease of differentiation gone wrong. Cancer cells often exhibit abnormal differentiation patterns, failing to mature into specialized cells, and instead proliferating uncontrollably. Understanding the molecular mechanisms that govern differentiation is crucial for developing novel therapies for cancer and other diseases. Section 8. The future of differentiation, stem cells and regenerative medicine. The field of regenerative medicine stands at the cusp of a revolution, fueled by the promise of stem cells, those unspecialized cells with the remarkable ability to differentiate into various cell types. These cells hold immense potential for treating a wide range of diseases and injuries, offering hope for tissue regeneration and functional restoration. Stem cells can be derived from various sources, including embryos, adult tissues, and even reprogrammed adult cells, known as induced pluripotent stem cells, or iPSCs. These iPSCs, generated by reprogramming adult cells back to a pluripotent state, circumvent the ethical concerns associated with embryonic stem cells and offer a personalized approach to regenerative medicine. One of the key challenges in regenerative medicine is directing stem cell differentiation towards specific lineages. Researchers are actively investigating the intricate signaling pathways and molecular cues that govern cell fate decisions, aiming to develop robust and efficient methods for generating specific cell types for therapeutic applications. Section 9. A fundamental process. The enduring significance of cell differentiation. Cell differentiation, the process by which unspecialized cells transform into specialized cells, stands as a cornerstone of developmental biology. It is the driving force behind the incredible diversity of cell types that make up complex organisms, from the intricate network of neurons in the brain to the contractile fibers of the heart. This remarkable process, orchestrated by a complex interplay of genes, signaling pathways, and epigenetic modifications, ensures that cells acquire the specific characteristics and functions necessary for their roles in the grand scheme of life. Understanding cell differentiation is not just a matter of scientific curiosity. It is crucial for comprehending the development and function of living organisms and for addressing a wide range of human diseases. The field of regenerative medicine, fueled by the promise of stem cells and our growing understanding of differentiation, holds immense potential for restoring damaged tissues and organs, offering hope for a healthier future. As we continue to explore the intricacies of this remarkable process, we embark on a journey of discovery with far-reaching implications for the future of medicine and our understanding of life itself.